So I'm going to give, for those of you who, I gave a similar talk a couple of years ago about stem cells and what we can do with stem cells relative to Smith-Lemli-Opitz. So I'm going to give a few introductory background slides about what stem cells are, different types of stem cells, not as extensive as a couple of years ago, and then get in more to our work that we're doing in my lab in South Dakota, using some induced pluripotent stem cells that we developed from SLOS kids back when I was a postdoc with Danny Porter's lab, and I'm continuing this work now. So my lab really has three main goals related to looking at how DHCR7 affects development and function. And we're using stem cell models to get at these questions. Number one, what are the developmental consequences of DHCR7 mutation? Number two, what are the functional consequences? And number three, can we develop therapies that can impact patient symptoms and improve quality of life? And I think using these induced pluripotent stem cell models that I'm going to talk about, we can really get at some of the specific signaling deficits that are downstream of the cholesterol biochemistry that we're all familiar with, to help us try and develop some therapies for some of the kids. So just a few slides about what stem cells are and what stem cells do. There's a lot of stuff in the news about stem cells and in the literature. So a stem cell really does two things. A stem cell is a cell type that can self-renew, so it can undergo different rounds of cell division to create a copy of itself, more or less, to keep itself in a stem cell state, and it can also differentiate. And when we say differentiate, it means a stem cell is becoming specialized. It's becoming something that has a cell type that has a specific function. So something like a neuron or a cardiomyocyte that beats, a neuron that fires action potentials. So something that has a really specialized function. I break stem cell groups up into four different types: fetal, amniotic cord blood stem cells, adult or tissue-derived stem cells, embryonic stem cells, and then really the focus of my lab are these reprogrammed or induced pluripotent stem cells. I'm just going to touch on adult stem cells and the iPS model, since those are really the two that would be most applicable to Smith-Lemli-Opitz. So adult or tissue-specific stem cells are found in virtually every organ of the body with the exception of the heart. So there are stem cells within the brain, within the intestines, within the bone marrow, fat, and this is how organs repopulate cells that are dying or need to be replaced. So this is constantly going on within the embryo, within kids, within us as adults, just usually at lower levels as we get older, for most tissues anyway. And these stem cells are going to be in an area where they're protected. We call it a niche, where they can maintain the stem cell state. So there are different stimuli that will keep them as stem cells, so that these populations of cells don't deplete themselves, so tissues and organs don't age out too quickly. Adult stem cells are typically multipotent, so neural stem cells can become neurons, for example. Stem cells from the bone will differentiate into bone and other tissues of similar origin. We don't usually think about stem cells, a brain stem cell becoming a cardiomyocyte. But an issue with most of these adult stem cells, with the exception of things like fat and stem cells from the blood and bone marrow, these are really difficult to obtain. So thinking about trying to get neural stem cells, isolating them out, you'd actually have to go in and pull them out of the brain, out of brain tissue, and that's obviously not applicable. Embryonic stem cells, so these are really primitive stem cells that are pluripotent, meaning that they can become virtually anything within the body. They can grow and self-renew indefinitely, and this isn't really true for other like adult-derived stem cells for the most part. We can isolate these cells out and grow them in the lab for a really, really long time, and we can use them for a lot of studies in the lab to try and understand how tissues develop, If we take a gene and disrupt a specific gene, how does it affect the development of a particular cell type or tissue? But these cells have ethical considerations if we're talking about human embryonic stem cells. There's limited availability. There's only so many lines, so many of these cells that labs across the U.S. can work with. Also, they don't typically recapitulate any disease. They're usually just from unaffected individuals. And they also, if we're thinking about down the road using stem cells as a cellular cell therapy, these cells will induce an immune response, like going from myself to anyone else in the room. So this kind of leads me to what my lab does with induced pluripotent stem cells. So with induced pluripotent stem cells, this is an artificial stem cell. This isn't something that's in anyone here. What we do within the lab is we take cells from an individual. We can usually use fibroblasts from like a skin punch, or we can take blood and reprogram blood. And so we can actually reverse development with these iPS cells. So typically, as a stem cell becomes more specialized, you kind of think about it like a ball rolling down a hill, and it has to make a choice between going from one valley to another until it gets down to whatever cell type it's going to become, whether it's a cardiomyocyte, a neuron, a fibroblast, whatever. With this iPS or induced pluripotent stem cell technology, we can actually reverse this process and take a cell from anyone in the room. It'll carry the same genetic material that is in anyone in the room, reprogram it back. So if there's a particular mutation that we want to study, sometimes it's hard to do that with fibroblasts, for example. So we can take it back to an iPS stage and then study certain cell types we want to look at. So iPS cells are kind of different versus other stem cells in that they have a lot of potential, this pluripotent potential where they become virtually any cell type if they're given the right conditions, like a human ES cell, but they don't have the ethical considerations. There's nothing related to human embryos or having to get oocyte donations for some kind of like nuclear transfer cloning. We can derive them at this point from virtually anyone, usually with a blood draw these days. But all of the Smith-Lemli-Opitz lines that I developed in Dr. Porter's lab and I'm working on now were actually from skin punches. We can use any cell type that is dividing, so fibroblasts, different cell types from the blood. And these models allow us to study a human disease outside of the patient. So we can really get down to how a patient's specific genetics is leading to different cellular phenotypes. And down the road, these cells should be able to avoid some kind of immune rejection. If you could take a cell type, reprogram it back, and then transplant it back into an individual, we should be able to avoid a lot of the immune rejection and suppression that usually has to go on with stem cell transplants. So these aren't a perfect model, but they are a good model for a lot of diseases, and I think for SLOS. So when we make these, they'll have the same genome, the same genetics that the individual who gave us the cells carries. But it also isn't just your gene of interest. Like if we're making iPS cells from two kids with Smith-Lemli-Opitz syndrome, they have two different mutations. The iPS cells will have those mutations. But if there are other genes that are mutated that we don't know about, those are also going to be within those cells. So it's kind of a confounding factor that we have to think about as we're doing experiments and assays with these cells. We can grow them up and expand them to billions and billions and billions of cells. It's not super fast, but it works very well. And we really don't want to study just the stem cells. We want to take the stem cells and then push them to a different cell type that we are interested in, whether that's neurons or astrocytes or inflammatory cells or cardiomyocytes, whatever. But there are issues with, this doesn't happen 100% of the time efficiently, so we have to come up with some interesting ways to get at specific populations of cells. So we can look for cell type specific effects, which is great. We can use pretty advanced differentiation assays to organize these cells into a spatially organized system. I'm not going to show it today, but we can actually do these assays in 3 dimensions and create structures with lots of different cell types together that look like a little mini organ that we can use to study development. But this is a really time-consuming and a low-throughput process. Has anybody in here heard of CRISPR and gene editing, probably in the news a lot? So we're using this a lot in my lab, using CRISPR to gene edit iPS cells, including the Smith-Lemli-Opitz cells, to try and correct the genetic mutation and see how that affects some of the cellular phenotypes that are relative downstream to the DHCR7 mutations, but also using the gene editing to try and track how specific proteins and things are affected by DHCR7. And as you can imagine, these studies are really challenging technically, but also financially. They're very expensive and they take a very long time. So it took me and Denny's, when I was in Denny's lab, it took us several years to develop the models, and then several more years to actually find something that was interesting. So these are really long studies to do. And as we grow these in the lab over time, there are a lot of controls and things we have to do to make sure that they stay stable over time, because they will pick up different genomic changes that we have to be on the lookout for. So what can we do with these cells? So we can take a biopsy or blood draw from an individual, grow up these cells in the lab, reprogram them back to this iPS state. So for some different conditions, mainly diseases of the hematopoietic system, things like leukemias, there are labs who are trying to take the make iPS cells from an individual, and try and correct the mutation with something like CRISPR, and then transplant those cells back into an individual. Now, this is a really focused technique if there's one single cell type that needs to be fixed, and you know what that is, and you know exactly what you need to do. For something like Smith-Lemli-Opitz, where it's such a broad spectrum disease, what my lab is trying to do with the iPS cells is differentiate them into different cell types and use these as a model in the lab that we can study the human disease without using mice or rats or fibroblasts, with the goal of we can understand the disease and hopefully get at some different therapeutics down the road. So these are the Smith-Lemli-Opitz cell lines that I generated when I was in Dr. Porter's lab, and these are the cell lines that my lab is continuing to work on now and characterize and do some different methodology on. So we have cell lines from kids who span the phenotypic spectrum of Smith-Lemli-Opitz and carry some of the most common mutations that we see within the kids, including the T93M, amino acid change, and then this G to C variant that causes a frame shift in one of the alleles. So, and we grow these. In order to make these cells, it takes about a month to generate the iPS cells. So it's fairly quick. That's quick from the perspective of a lab experiment. But the issue becomes down the road when we have to try and characterize and validate what these cells are. So we have to do a lot of different assays, looking for different proteins they express and make sure that they have the right, they carry this mutation that we expect them to. We have to do karyotyping on the cells, and then differentiate them out and make sure that they can form different cell types like cardiomyocytes. You can kind of see this thing beating in the dish. And we can also inject them into mice to make sure that they can form different germ layers in the mice. So this process usually takes about a month to make the cells, and then if things go really well, this takes about 6 months, sometimes a year or so. And so all of the IPS lines that I was able to generate in Dr. Porter's lab, we compared and validated against control lines that we were, and we've been very happy with the IPS lines we were able to make with Smith-Lemli-Opitz, from Smith-Lemli-Opitz patients. So the major finding I had when I was in Dr. Porter's lab was we identified this novel loss of a protein called beta-catenin downstream in these IPS lines that have these DH07 mutations. Really, the most important thing here that you should take away from this is what we're doing here is looking at the expression of this protein beta-catenin, and really I just want you to notice that it's different here between this panel and this panel. There's less here, it's much more stable here, and we can actually stabilize this with a small molecule. So this pathway, this Wnt pathway, is a really critical developmental signaling pathway that controls a lot of different tissues and how a lot of different tissues develop. It controls stem cell function, how stem cells self-renew, what they decide to differentiate into. And I think that this defect in Wnt signaling probably helps explain a lot of the developmental malformations that we see within the SLOS patients. But kind of the consequences of this disruption are really not clear. And so what we figured out was that in the cell membranes of these cells, you see these little green structures are cholesterol, and in Smith-Lemli-Opitz, where cholesterol should be, there's the 7-dehydrocholesterol that's within the membrane. And this signaling pathway, this Wnt signaling pathway, is like a complex of different proteins that have to come together in order to function normally. In cells that have the 7-dehydrocholesterol in the membrane, this complex of proteins doesn't form well. It's very unstable, and that instability causes the pathway to be suppressed. And I think that's that's causing a lot of the developmental issues in Smith-Lemli-Opitz, but it's not the only thing that's happening. So what my lab has been trying to do is better characterize how critical this Wnt pathway is to the neurodevelopmental aspect of Smith-Lemli-Opitz. So we've been taking our IPS cells from the kids, and here I'm just confirming that we're seeing the same kind of Wnt defect that we saw previously in the Smith-Lemli-Opitz patients. We see the same biochemistry that we see within fibroblasts from the kids. So what we try and do in the lab is to suppress we can environmentally control the suppression, much like of the biochemistry, much like we try and do in the clinics with cholesterol supplementation. It works pretty well in the lab, where we can add cholesterol to the cells. They'll take it up, it'll suppress the biochemistry, and then we can do our assays down the road. So we get pretty good suppression. We take the cholesterol away from the media, and we see the red increasing, which is the 7-dehydrocholesterol, and the cholesterol decreasing in the gray. So we can recapitulate the biochemistry pretty well. One new thing that we're finding in the lab is, yes, there is this decrease in this Wnt pathway signaling, but there's also a change in how the proteins downstream of Wnt are actually localizing. This is a little on the sciencey side. But this protein that we lose, beta-catenin, which is so critical for development, has two different functions. It functions at the periphery of the cell and allows cells to bind together, but it also goes into the center, into the nucleus of cells, and causes cells to transcribe and make new DNA. So it has two different functions. So initially we had found that we see this protein gets lost. So we just kind of assumed that we're losing both the peripheral changes and also the nuclear changes, the genetic changes. What we actually see in the lab is we see an increase, even though the overall levels are going down. The way the protein is moving around the cell is very different, and I don't have any idea why that is at this point, just that it happens. So we get more of this protein in the nucleus, which I think is driving some of these early developmental malformations that we see in our cell lines. And if we inhibit that, we can maintain the cells as stem cells, and has a pretty significant effect on our cell types. So Wnt signaling is much more complicated than we had first thought. Another thing we found that's new. I mentioned this Wnt pathway is critical for tissue development and deciding how a stem cell what cell type it's going to turn into. The level of expression of this protein will define more or less what a stem cell decides it's going to become. So if it's going to become a neuron versus an astrocyte, if the levels change, then that will affect the ultimate outcome. So what I'm showing here on the top are we've taken stem cells from one of the Smith-Lemli-Opitz IPS lines and a control. You see there's a lot of gray, there's a lot of red, there's a little bit of green in here, but just ignore the green for now. Hopefully you can appreciate there's a lot more green on the left than on the right. A lot more red on the left than on the right. So the red is actually a protein that labels astrocytes. So when we take the stem cells from these cell lines and we can push them towards different cell types we want to look at in the brain, we mostly think about neurons and astrocytes. So neurons are what we think about are primarily the functional cell type of the neuron that causes the brain to function and form synapses and motor control, thinking, memory, all of that. Astrocytes are typically thought of as like a support cell that helps the neurons to function well. And what we see in the Smith-Lemli-Opitz is there's not a lot of red here. So we see a suppression of this astrocyte formation in the Smith-Lemli-Opitz IPS cells. And this is something that hasn't been characterized before. And this is pretty unique even to neurodevelopmental and IPS models. That this defect in astrogenesis. And concomitantly, we should have certain levels of neurons and astrocytes, and if you upset this balance, then that's going to upset the functional activity of the neurons within the culture. This is partially Wnt dependent. If we stabilize this pathway that I identified in Denny's lab, then it has an effect, but it's not 100% effective. There's a lot of different things going on as we go from these pluripotent stem cells to a neural stem cell to immature neurons to mixed cultures of neurons and astrocytes and glia that you would find within the brain. So here I'm just kind of summarizing a lot of data, looking at genetic changes in different genes as we go from one cell type to another. So this Wnt pathway seems to be critical for all of these different transitions, but there are a lot of other pathways that are affected, that affect neurodevelopment, and it's really cell type and cell stage dependent. So as my graduate student likes to say, there's a lot going on here. And so we're still trying to get a handle on different cell type effects. To get at more of a functional effect, and this slide is still messed up. We're also taking our IPS models and trying to look at neurites outgrowth. So some previous work in Dr. Porter's lab a while back had identified that if you took neurons out of a mouse that we used to model Smith-Lemli-Opitz syndrome, that these neurons would have really long projections. And that would be expected to affect neuronal function. So if there would be a way that we could use our IPS models with different patients with different mutations in DHOS7 to try and model this neurite developmental aspect, then we could use these to look for different pathways that are affected, try and identify drugs that could affect remodeling of this process, and maybe get it more of a functional benefit. So what we can do is we can take our stem cells, we can push them to become neurons, and then we can use an imaging system, it's an automated imaging system that will quantify how many of these projections come off, how long they are, how thick they are, how much they branch, and these are all different measures of how functional the neurons should be. Thankfully, we're able to recapitulate we see the same kind of phenotype in the IPS model that we saw, that Dr. Porter's lab saw in the mouse, whereas these neurites, these projections that come off, are much longer in the Smith-Lemli-Opitz. Here I'm just showing some data from one of the patients with the T93M mutation and then the frame shift on the second allele. We can suppress this if we give the cells lipids in the media, so they'll take up the lipids, suppress the biochemistry, so that kind of gives us proof of principle. But also if we use this drug that affects beta-catenin activity and Wnt signaling, we can also suppress this. So this was good that we were able to see the same thing in the human cells that were previously identified in the mouse, and also that there is something that will affect the remodeling of these projections. But we're also able to do measure a lot of different parameters related to this. The SLOS cells also have a lot more of these neurites that are coming off. So in principle, that could be a good thing, or it could be a bad thing. There's a developmental paradigm where, as a neuron develops, depending on what the neuron comes into contact with, they'll form all of these neurites, and then as they get different stimuli, they'll prune and remove some of these neurites to become more functionally mature. At least in the SLOS, there may be an issue with how these cells are actually pruning off these neurites, and this might be a good readout for functional activity. And then this ramification index, what this is actually measuring is giving you an idea of how well these neurites are projected to contact and affect other neurons around it, to get at kind of how the neurons are going to talk to each other and function within a system. And that's actually impaired in the Smith-Lemli-Opitz. And so our targeting of the Wnt pathway has some benefit, but not consistently across the board. So we're trying to use these kind of methods to look at more functional readouts of using these IPS models. So on the left, we're doing some fluorescent and microscopy imaging, looking for formation of synapses, or we're looking for proteins that we know are present in synapses. So these are going to be the functional, where neurons and astrocytes functionally interact to fire and release things like neurotransmitters that are going to control brain and neuronal function. So we can see that the Smith-Lemli-Opitz cells are definitely expressing a lot of the proteins and things that are going to be important for synapse formation. We're also using things like CRISPR to tag proteins that we think might be important for neuronal function, and look at how these proteins move around in real time when we inhibit or activate different pathways or affect the biochemistry with things like cholesterol. And we're also collaborating with labs outside of South Dakota to look at network level activity. So this is actually calcium imaging, where we have neurons and astrocytes within a dish, and all the green firing is calcium flux. As neurons are generating action potentials to fire normally, this calcium flux is a critical readout. So you can see the green flashes everywhere where the Smith-Lemli-Opitz cells are forming action potentials. So we're trying to quantitate how that compares to controls, but also when we add in different drugs, how does that affect? How is that affected? Okay. So just to wrap up quickly, using these IPS models, we've identified that there are some significant developmental effects downstream of the DHCR7 mutation, and we kind of expected that. The Wnt deficits, I think, are a significant contributor to a lot of the developmental issues and malformations that we see, but there are also a lot of other pathways at play. The astrocyte numbers, reduced number of astrocytes, I think is very interesting in the fact that less cells being able to support the neurons are going to affect things like how well neurons form synapses, how functional synapses are, nutrient transport from astrocytes to neurons, and also things like neurotransmitter recycling. And so we're using these neurite assays to collect data and develop screens to use like a non-biased, like automated quantitation system, so that we can get at functional deficits and hopefully identify some small molecules and drugs that might work. But we need a lot more data as far as looking at multiple cell lines that kind of span the spectrum. These neurite assays are pretty labor intensive, and so we're really at the infancy stages of this, although the data is very interesting, but we need to do this with a lot more cell lines to get at some of these functional studies. So I'll just thank the people in my lab. Really thankful for the foundation for giving us a pilot grant last year that helped get some of these neurite assays up and going, as well as my funding through NIH, the families who have donated tissue for the studies. None of this would have been possible without the support of the families for the research, and Denny for the training and for generating the IPS cells. So, and afterwards, if anybody has any questions and want to send me an email, please send me an email and ask about these IPS studies. So, and I'll take questions if anyone has anything. Okay. Thank you. Oh, sorry.
I was just going to ask, are you, with all this testing and everything that you're doing, is this going to help a drug company to know what drugs to maybe test, or are you just trying to understand SLOS more?
Hopefully it does both.
Okay, great.
The idea for the neurite, the imaging studies, would be to find something that's going to find some kind of a drug or compound that will affect how these processes remodel and shape themselves, to get them to a point where they're more like unaffected individuals. So ideally, yes, we would find something that would either be maybe already FDA approved or in clinical trials for Alzheimer's disease or Parkinson's disease, where they're also looking at these different proteins that affect how neurons remodel, and it could be something that would be applicable to SLOS. That's the goal.
It just seems like quite a process.
I'm sorry?
It seems like quite a process that I didn't understand how long it takes to just do one little thing.
It's a very long process. These neurite, the assays I showed you where we're trying to plate these cells out and quantify them, it's taken us a couple of years to get this to a point where we're comfortable with the consistency, that we can use it for something like take a small drug screen, and we can use things that have worked on other diseases, at least in the lab, and then apply it to SLOS. So it is a process,
Wow.
but it's going. So, thank you.
Hi, I have a question.
Hi.
I apologize. I had to leave during
That's
your
okay.
talk, and I missed most of it. And I'm a neuroscientist by trade, so
Okay, that's okay.
Sorry that I missed it. But for future families that are coming into the group, is there an advantage to keeping cord blood of our own to do more personalized medicine for to get access to our own stem cells?
Yeah. I mean, I guess my issue with cord blood, you can bank it away indefinitely. Different companies, different places do a better job of that than others. It's not cheap. It's not cheap to do. Most of the cord blood, when we think about cord blood, we're thinking about some kind of a transplantation study where you're taking the cord blood and then putting it back in to the individual down the road or to a sibling. And I really don't know. I can't foresee at this point, at least for the neurological aspect, how cord blood is going to be a huge benefit for Smith-Lemli-Opitz. It could be used it could definitely be a good model to use down the road to maybe confirm some different things. But as far as a regenerative or repair mechanism, I'm kind of skeptical of cord blood at this point for SLOS. But I would also kind of defer to some of the clinicians in the room as far as what they think related to cord blood and it as a possible therapy for SLO.
I'm just wondering about access to stem cells specifically.
Say that again, the active stem cells? No, that's okay. So you're about the stem cells.
Going back to my CDAT conferences,
Yeah.
I'm just wondering about the access to stem cells specifically from the cord blood, if there's an advantage to having your own individual stem
Yeah.
cells versus,
Sure.
Yeah,
Yeah.
versus adult stem
Yeah,
cells
so
or something.
so there are a few different rare neurological diseases where they're using different stem cell transplants, like Krabbe disease. They're using stem cell transplants to treat the neurological aspects of Krabbe,
Yeah.
and it does a fairly decent job, but it's all about support. You're not actually fixing anything that's there.
Right.
And it's a short-term effect. It doesn't last forever. So yes, there could be potential, but I think there would need to be, before I would say yes,
Yeah.
I think we would need to do like some more basic research studies where we're taking maybe cord blood and putting it into a mouse, or if we had a large animal model for SLOS, we could put it in a large animal model and see how it affects that. But that's kind of where I stand.
All right, thanks.
Okay, sure. Anybody else? Okay, thank you.